All right. So I have a question um, for the whole panel. Um, the cardinal rule of a trial is uh, one trial, one hypothesis, one question. Um, how do we handle the complexity of all of these um, issues of generalizability? Um, whether it's does the effect size, um, the average effect size apply to individuals or not? Um, and in Constantine's presentation, um, does the effect size change over groups of patients? And in ESA's, we begin to get towards some of the issues that Chris brought up, which is um, when we have two different data sets, one from healthcare delivery or epidemiology or um, a longitudinal study without intervention, how does the baseline condition relate across groups? Um, and from Chris's perspective, uh, I think I take your point that redesign of how we do business is important, but you ended with, we don't know the precise value of redesign, so we have to look at our data for many of the issues that you brought up. So how do we get at all of these issues in some organized way when the main job of the trial is to answer one question about the hypothesis and all of these other things become secondary?
Can I offer one thought, Dan?
Yeah.
So just a way to weave in a little bit this concept that we don't quite know how to do it right yet. This is where the discipline of clinical and translational science comes in, studying how we do this. And so I would posit if we design a clinical trial that is to answer a clinical question, we can embed inside of that trial different approaches to participant recruitment and answer a question about which approach leads to a more diverse population. And that doesn't necessarily undermine and shouldn't undermine the trial question, but it does begin to answer the question of how do we optimize design for diversity at the same time as answering the clinical question.
So an embedded variance within an overall trial.
I believe that every trial that we do should have as many embedded questions within it that do not undermine the clinical question, but really help us to make the next trial we design as effective as it can be.
Okay. All right, let's go back down the size of the question to Isa. What do you think about my question?
So I have to say, my talk was very classical in that sense. There was one estimand, you can do one test. I just provide a faster way, more efficient way to get an answer to the same question.
You
But
also told
instead,
us, let me interrupt for just 1 second. You also told us that there are many ways that the estimand fails and that it's only in situations.
Yeah. So, I mean, one setting in which I've come across, I think the issue you're asking about is very related to Chris's talk. Some of our work is about observing the trial, seeing, even if the trial itself is designed for representativeness, to use Chris's terminology, it won't be representative of the next population where I intend on drawing inferences from that trial. So although I, of course, 100% agree, it's good to have a more diverse recruitment process, or a recruitment process that aims for diversity among the goals. I think there's always going to be the need for post-processing to the extent that the trial results are going to be applied to a different population. And so there, I think a question like the one you're posing, Dan, arises. You have two objectives. One is to do efficient testing of hypothesis or estimation in the trial population, and to also learn about representative group. And so there, I think there is a lot of fertile ground for translational work, taking ideas from experimental design with multiple goals and trying to basically power the study to do both. We've taken like a teeny step in proposing methods where if you can control the probability of participation, as is the case, for instance, in cluster randomized trials, you control the probability of selecting clusters from a roster, you can jointly improve representativeness and efficiency in the trial. And so I think there's a lot of space for creativity there, but I don't know too many people working on that problem.
Okay. I think that's a good answer. Let's go through the other two speakers, and then we have a couple of questions here. But yes, the references will be available in each of the talks, and the R packages or other packages will be available with the talks on the website. And we'll get to Frank Harrell's question in a minute. Konstantin, do you want to go next?
Yeah, so I can answer for how I see your question in relation to what I presented. I think that the powering of a trial for the sorts of exploration or identification that I described would probably, if done explicitly, would probably require or increase the sample size by some amount that will differ from trial to trial. But that's a different question or issue than to say if you do design a trial in which you measure and you plan to measure many covariates, how would you go about finding suggestive subgroups that you would then in a separate maybe or separate plus the current trial data would plan for nailing down the this additional question.
Okay. Ravi?
What's the question, Dan, for me? Is that about sample size or?
Well, the question is, trials have a single hypothesis and a single goal. And when we look at whatever they are, subgroups, secondary effects, process variables, we're, as Konstantin pointed out, we're not powered. But how do we do this in an organized way so that we create knowledge about all these other interesting questions? I mean, is it as simple as the way we currently in government-sponsored processes pay for trials, that we tend to pay for the primary analysis, and we don't fund paying for all of the other secondary analyses, like the whole process analyses that Chris, I think, correctly brought up, that show us that we don't always design our trials right. And I think Isa's presentation showed some of the flaws and assumptions in trials that became conditions for the pooling of one data set and another. So your thoughts on that issue? How do we benefit from these secondary analyses?
Right. It's just I saw Frank's question about the reliability of these methods, or maybe the stability of these methods, and how sensitive they are to the lack of information, perhaps, because you're doing very ambitious models. It's a tough one. I mean, the way I've thought about it, not perfectly maybe, is that it's more like you have all this rich data available, and to report only an average treatment effect. I understand that is how the study is designed, and it is designed to answer that question very well. But now I'm just saying that, well, can we answer more questions than just that?
Yes.
Right? And I'm not saying that the answers would be trustworthy all the time, or maybe even a few times, but we need to try these things out, and and have maybe like validation studies to follow up if there are some startling findings that emerge from these exploratory analyses. Perhaps we take the next step and see if they are really serious, or they're just artifacts of the modeling exercise, And until we get some practice doing these things, I know that in the subgroup world, there has been a lot of skepticism that, subgroup analysis that have been done in randomized trials almost never pan out in follow-up studies. Richard Pito and others have really blasted these subgroup analyses before. But I also feel, on the other hand, that just not doing these things is also irresponsible, to do a large trial and not do anything else with the data, I think both are irresponsible. Like I used to say like the FDA perspective versus the PCORI perspective. The FDA perspective is focused on the drug. The PCORI perspective is focused on the individual patients. Do I benefit from this treatment? I mean, talking about representation and all that, right? But I want to ask, I'm an Asian, I'm from India, I have some issues. But when I ask my doctor, do I need to go through colorectal screening? I'm a vegetarian and I don't know if anybody that I know of has colorectal cancer, And he says, well, this is the standard guidelines, I don't know what to tell you, So these are the types of things. I feel like unless we get some practice doing these things in real trial settings, we really don't know what to trust and what not to trust.
Yeah, it sounds like you got the average patient response to your thoughtful question. Maybe Frank Harrell wants to jump in with his question again. Frank, are you still with us? And can we open a microphone for Frank? There we go. Unmute yourself.
I hope I'm unmuted now.
You are.
Thanks for the opportunity. I tried to express it in the text there of the question, but the basic idea is that I've noticed a correlation between the lack of a sample size calculation and the trustworthiness of the product. And I've asked speakers who have proposed various subgroup or heterogeneity of treatment effect procedures, have they actually done a sample size calculation? Because they certainly didn't present it in their seminar. And they said they actually did, and they found out their sample was totally inadequate for what they're trying to do, but they wanted to present it anyway as a method. That's not very impressive. And I think a lot of things we try to do, keeping in mind that the clinical trials are usually undersized for estimating the average treatment effect. Power calculations are usually gamed with effect sizes that are bigger than the clinically reported difference. So, we try to push the data too far sometimes. So maybe someone can tell me I'm totally wrong.
I don't think so. I think those are pretty solid observations.
Yeah, I'd like to comment on this one, Frank, because I think that you're 100% right. And part of this is a little bit what Ravi was talking about, the difference between kind of FDA and NIH and PCORI. When we're looking at efficacy of a drug, we try to control the situation as much as possible. And heterogeneity of treatment effect is something you're trying to eliminate because you want to get as much signal over the noise. Whereas if you're going to more of a PCORI framework where you're really trying to understand effectiveness or implementation, what matters is not, does the drug work on average for a group of very homogeneous folk? You're interested in that individual treatment effect. And so in that context, the area where we are being very, very prolific in sample size calculations for differential or heterogeneity of treatment effect is in our large PCORI trials. And so that's the framework, Frank, where I am seeing it because the trial size allows for it. So I think that in the small efficacy studies, I would agree with you, and I would be challenged to believe a heterogeneity of treatment effect that didn't have a pre-specified power calculation. However, I think that as we get to implementation and comparative effectiveness, our trials should be designed to have the sample size sufficient to answer the heterogeneity questions.
I think those are great comments, and I just wanted to mention one aspect is you mentioned homogeneity in a clinical trial. I think heterogeneity actually works to your advantage in a classical clinical trial. So I think you really want to have a lot of heterogeneity, even though you're estimating an average treatment effect as your first job.
Can I also say something? What comes to my mind in relation to what Ravi said, which is it's irresponsible to promote something like a subgroup analysis as a finding, but it's also irresponsible to ignore it. And I'm not a clinician, but I remember seeing in a documentary that when people were trying to see what is the R square or whatever, or how damaging the total cholesterol was for heart disease, and they looked at many people, and it was a tiny village in Italy that was the mismatch to the whole pattern that showed that there is actually a good cholesterol too. And I raise that to say that I'm pretty sure there was absolutely no power for that little bump of the data to be significant as a diversion from the overall pattern. But people took the courage to look at that village in Italy, actually in southern Italy, and they found that that little village, they had very high cholesterol, but they had very low cardiovascular problems, and it was because they had good high cholesterol. Again, I'm not a clinician, so I may be getting it wrong, but I remember that very vividly. And again, my reasoning for saying that is that they would not have discovered that there is a protective cholesterol if they had said, oh, because this little bump in the data down there in Calabria or whatever has no power in our analysis, we're gonna ignore it. That's what I want to say. So I do agree with Ravi on that binary, not do this, but also not do the other extreme.
I can't resist mentioning that even though that bump was identified, it's never, ever been able to be actioned. So the treatments that have tried to raise HDL cholesterol have either not helped anyone or have actually hurt patients.
That's true. There are other examples. This one hasn't been actioned either, Frank, but some of that's the time constant for affecting designs of trials with new biology, and that's single patient in a village in Venezuela who showed the critical interaction of tau protein with amyloid in creating cognitive dysfunction in dementia. That probably would have happened on its own because of the football frontal lobe tau protein story, but there is biologic information sometimes in small numbers, but how to make it actionable is the question, and how to make it robust. Do you want to speak to the second point you put in the comments, because I think that's one way to make to at least evaluate the secondary analyses.
Are you speaking, Dan, of my comment on your question?
Yes.
Yeah, that was just a crazy idea because I think what happens in most trials, you don't have enough power to do something simple, and then you try to do things that are complex. Just doesn't work. And one of the things people try to do that's complex is they try to look at secondary endpoints that are low incidence. And but you might envision a study where you have a main endpoint that's got moderate power, but then you have a high resolution lab measure that you want to analyze as a secondary analysis. That analysis is going to have, tremendous power compared to the primary analysis. And those kind of secondary analyses are probably safer than the typical ones we see.
Right. We're actually doing that in a pharma type, but NIA sponsored study where the biomarkers have much more power than the clinical and patient oriented endpoint. Although Chris was a little negative on we're collecting too much data so that we can have something else to study later on. I think that there's an alternate hypothesis there, Chris, and that's that we're not pharmaceutical companies. We don't have a mountain of cash behind us to go study the next question unless we have evidence. And so I think there's a middle ground where we probably, it's the fundamental story of any science lab. If you just collect data and don't look at it intensely, seriously, and with an open mind to alternate hypotheses, you often move from question to question, but don't link the evidence that's within the data. And somehow we have to be able to take from our trials, which are such big efforts, more data about all the questions, admit that we don't know how to do the trials the right way, which I think came across very nicely in your presentation, Chris. But then ask questions of both the secondary outcomes, maybe the ones that are better powered, and of process outcomes. It seems to me that we have to look at our own data a lot more.
Having been the person who has flushed approximately 10 million biospecimens down the tube because they were sitting in a freezer for 30 years unused through multiple freeze thaw cycles, Dan, we collected a lot of data that we never look at.
Right, and that's a big crime, right?
But
Okay.
What's the benefit of looking more at the data, which I agree, by the way, if it's looks that you have thought about after the trial has been done? I mean, that's a provocative question. Huh? That's what I'm saying.
Well,
so I'm not sure where we left.
My trials, I haven't always thought of every question that I have after the trial is done.
Of course, and that's what I'm asking this because I'm not sure where the question about validity, some validity be gained and some scientific value being gained. Is there any such value gained from looking at the data in ways that you have not planned before? Do you have to have planned the power for every question that you will raise in your mind after you have planned the data? Is it illegal and it is stupid? What is it? If it doesn't have the power, I mean.
I think this is a really challenging area of philosophy. What is the value of a secondary analysis of a clinical trial data set for a variable that was not cleaned properly or monitored correctly? Is it valuable to look at? What is the benefit of using real world data to answer questions if it doesn't actually include the treatment effect of interest? These philosophical questions of what is the benefit are really important. I would argue that a clinical trial is optimized to answer the primary question. We can then add in the key secondary and exploratory endpoints, and we optimize for those as best we can. But after the fact, I would say that most of what we're doing is exploration and hypothesis generation that would require a subsequent prospectively designed assessment. Whether it's using existing data or trial data doesn't matter, but it would require a prospectively designed assessment of the newly developed hypothesis.
Yes, I agree with that.
Right. And that's my point, that we won't even do it if we don't plan in our trial plans to make some a priori assumptions. And I took from your talk, Chris, we ought to be making some assumptions about trial operations as well as the primary biologic goal of the trial.
If I may say something.
I have a question for you at the end, though. So I don't want
Sure.
you to leave, and I don't want whoever is staying to leave until I get my answer from you on a very important question.
I'll be very brief. And I know this is recorded, so I'm taking a risk. But basically, I wonder if sometimes these kinds of, ideas like, is it illegal? Should we allow people to do this? Maybe we're taking statistical methods too seriously in asking these questions. What I mean is that the formal models, I love that stuff as much as anyone, but they're not meant to replace thinking. They're meant to aid thinking. And so, I sometimes say rule number 1 is don't fool anybody. Rule number 2 is you're the easiest person to fool. Meaning, I don't think you can ban doing the analysis, but you have to have the cognitive preparation to avoid believing your work a little too much. And I think a lot of the subgroup stuff falls in that category. Underpowered, haphazardly done secondary analysis is the same. I'll stop here. If there's a question for me, I'm happy to.
Yes. I couldn't agree with you more. Chris gave you applause. I think everybody else was nodding their head. Embracing that we don't know everything and the alternate hypothesis is one way to save yourself. So you had a beautiful slide at the beginning with 3 points. And then you told us that Greek was a superior language. And then you told us that that because it's a superior language, your 3 points rhyme. So I would like to hear you sing your 3 points to end this very nice conference.
Not as superior as Sanskrit, Isa.
I have to say my name doesn't reveal it, but I was born and raised in Greece. My mother is Greek. My dad is not. And so I speak the language and I miss it. But we say ti thelo, ti echo, ti kano, which ends in an O type sound. And it means exactly what I put on the slide.
Okay,
Greek people and their language, Konstantin probably knows as well. We're a little too in love with it. So
yes. So one more time for all of us, just in Greek.
Ti thelo, ti echo, ti kano.
Tielo, tieko, ikano.
Almost like I would say.
Sounds like Vini, Vidi, Vici.
All right. I think we need to thank everybody for persisting. We still have 35 people listening. And Guyan, do you want to sum up?
I just want to thank everybody for all the effort and the time and really incredible talks. Very, very thought-provoking, interesting, thoughtful. So thank you very much.
Thank you all very much.
Thank you.
Thank you.